Welcome student. In the last lecture of our topic anomalous structure, we have discussed about anomalous structure arising before secondary growth formation and after secondary growth formation in dicot stem. Now we will discuss some structures which are there in monocot stem as well as in root of plants. So first we will discuss about monocot stem. One very important example is Dracaena stem. Okay, so Dracaena, I think so you all might have seen. It is a monocot plant. Okay, you can make out the difference by seeing the leaf structure. Okay, many other anatomical structures you can make out. So definitely, if it's a monocot stem, so when you will cut the section of this stem, what you will observe? Definitely scattered vascular bundles. Okay. So if a monocot, definitely it will have an epidermis. Okay. You can have hypodermis. So in hypodermis, after hypodermis, we will have ground tissue, and in monocots, as we all know that we have discussed before, that ground tissue is not differentiated into well-defined cortex, pericycle, endodermis, pith. So everything in general we call it as ground tissue, and in ground tissue we see lot of scattered vascular bundles. Okay, and what is the characteristic features of these vascular bundles? They will be definitely they will be conjoined. Okay, they will be conjoined, they will be collateral, okay, but they will be closed. Closed means no cambium. So in between this vascular bundles, you will find phloem on the outer side and xylem on the inner side, as well as both the secondary tissues will be on same radii. So that's why it is conjoint and collateral. And then this will be closed because no cambium. So if cambium is absent, definitely there won't be any secondary growth, and that is applicable in many of the monocots, that you will not find secondary growth formation. But in Dracaena, if you would have seen that plant, you could observe that it has somewhat thick stem or bigger stem if you compare it with other monocots like wheat, grasses, rice. So the stem is quite thicker. It is because of the presence of secondary growth or presence of secondary vascular bundle. Okay, but when this vascular bundle formation will take place, it is not in the normal way as we find in normal dicot stem. It will behave in a different way, and their arrangements will also be different. So let's try to understand what you will see. See, when you will see the primary or the initial stem, you will see the structures like this. Okay, vascular bundles will be surrounded by a bundle sheath. Okay, so that all features will be there. Now, when this will have secondary growth formation, you will see a different structure. Okay, so you will see formation of secondary vascular bundles which will be embedded in the conjunctive tissue. Now, these secondary vascular bundles will be found in the outer region of this Dracaena stem, and since it is going on secondary divisions and it has somewhat a bigger diameter of stems, somewhat thick diameter, you could also see some cork layers, some sort of active cork cambium will be there. Okay, so that also structures you can observe, but let's try to focus here what is in the vascular region. So this will be the primary vascular bundle, and this will be the secondary vascular bundle. Okay, and this will be the conjunctive tissue. Okay, so conjunctive tissue will be basically thick, okay, and it will be sclerenchymatous. Okay, so when we will stain them, we will see that this tissue is also staining pink in color, and in that between you will see secondary vascular bundle. Okay, you won't definitely see any endodermis or pericycle because it's a monocot stem. Now, coming to the structure of this vascular bundle, see, this is also very important. It's not normally, its formation is not normal, and its organization is also not normal as the normal vascular bundles. It is amphivessel in nature. See, before that also we have discussed that amphivessel means phloem is surrounded by xylem. Okay, so phloem is surrounded by, suppose this is the phloem, okay. If this is the phloem, it will be surrounded by xylem. If we make a cellular diagram, we can make it as small group of cells are there in the center, okay, and then it will be surrounded by thick lignified cells of xylem. Okay, so this is xylem and this is phloem. See, when you will see the structure of this vascular bundle, it will appear almost rounded in structure, whereas if you try to find out some difference between the primary vascular bundle and secondary vascular bundle, you can see that the primary one will be slightly oval in shape, okay, and in primary vascular bundle, what do you have? You have phloem on the outer side, okay, so definitely xylem will be on the lower side and you will have phloem on the outer side. But yes, this is also surrounded by a bundle sheath. So definitely in this it is also surrounded by a bundle sheath. So that bundle sheath will be sclerenchymatous in nature. Okay, so this is the primary vascular bundle and this is the amphivessel. Okay, so this type of structure you will observe. Now, how these vascular bundles are getting formed? So, in this case also you will see that some of the outer layers of the cortex, okay, since these vascular bundles are present on the outer layer, so some of the outer layer of this cortex will become meristematic, okay, they will form like, they will start behaving like cambium. So, what will happen? Now, if suppose this is the cambium, this cambium, what it will do? It will cut on the outer side parenchyma. See, it is not going to cut xylem on one side and phloem on other side. The behavior of this cambium is entirely different. This cambium will cut parenchyma on outer side and alternately and phloem and xylem both it will cut on the inner side, and phloem and xylem on inner side. So, let us see how it happens. See, it is cutting phloem parenchyma, and on the inner side it will cut parenchyma and xylem in patches actually. It will and phloem and xylem on inner side with parenchyma in patches. Okay. So, it is cutting. First of all, it will start making, suppose xylem, it will make xylem. Okay. It will cut xylem. Okay. After some time, the activity of the cambium will be reversed. Normally, it is cutting parenchyma, but at those locations where it was cutting parenchyma, it will start cutting phloem. Okay, you can see. Okay, and then again its activity will be reversed and it will cut xylem again. So, on that same place, so that means now phloem will be surrounded by xylem. So, this will be xylem and this will be phloem. So, in this case, in this way, this amphivessel vascular bundles will be created. Okay. This is cutting actually parenchyma, but later it will be sclerified and it will be converted into thick cells. Okay. So, and that is why when you will see the cross section, it will appear also pink in color, and they are more compact cells as compared to the parenchymatous ground tissue. So, when you will see the structure, see this is also a section which I have prepared for you to understand. See, this is also the TS of Dracaena stem, in which you can see the vascular bundles, lot of primary, these are all primary vascular bundles. Okay. So, here you can see, this is all vessel, you can easily make out. So, it is vessel, so it is xylem. So, xylem will be on the inner side towards, and above it will be phloem. Now, you can also see pink color boundary of this cell. So, definitely this pink color boundary of this cell is definitely a bundle sheath. Okay. So, a lot of vascular bundles you can see in Dracaena, and they are having boundaries, and later see many of the pith cells will also start becoming sclerified. See, that also I have discussed before that in monocots, although many of them has very weak stem, but the several monocots like bamboo and palm trees, they become very big and they are very strong, and they are rigid structures. So, because of the presence of large number of vascular bundles and sclerified vascular bundles, large number of sclerenchyma, fibers and all the sclerified tissues are present. So, because of that they become so thick. In Dracaena, however, another type of vascular bundles also originate, which is secondary vascular bundle, okay, which is different from primary vascular bundle. So, here you can see secondary vascular bundle. See, here also you can see the structures which is not very clear, because in this section might be it is not very clear because these stems are very difficult to cut. Okay. So, but yes, they are also appearing as rounded structures. Okay. You cannot make out much difference between primary and secondary tissues, because they are also appearing rounded structures covered with pink colored encircling that xylem appearing as pink colored structure. But yes, the difference you can make out is about the conjunctive tissue. You can easily see the conjunctive tissue, which you can see that it is different from the parenchymatous tissue. You can see a thick wall, tightly packed tissue. So, you can assume that the vascular bundles which are found in these tissues are the amphivessel vascular bundle. Okay. So here also you can see the cellular diagram where you can see these are the normal primary vascular bundles where you can see the bundle sheath. Okay. Here also you can see this is the phloem and this is the xylem. Now in the sclerenchyma, which is the conjunctive tissue, they will be converted into sclerenchymatous tissue. Okay, you can see the xylem and this is the phloem. Now above this a cambium layer will be formed and this cambium is not in the center. Okay, it's just cutting out parenchyma on the outer side and all the xylem and phloem tissues on the inner side. So this cambium it actually appears to be on the outer side of the secondary tissues. Then you will have a cortex, and because it's also undergoing secondary growth formation, having a thicker and wider stem, so it will also have cork and cork cambium formation. Now coming to anomalous structure in dicot, root. See, root also exhibit lot of anomalous behavior, so that also some examples we will try to understand. Now one example is about sugar beet or beetroot, which is Beta vulgaris. See, you all would have seen it. See, it's an underground root which has been modified. It's a storage root. You can see that a lot of food materials are there in that root, and it has modified into a very big bulbous structure, and lot of materials are stored in this. So you can see when you will cut this, you will see that lot of ring-like structures are there. Okay, these rings are actually the formation of secondary tissues. Okay, and lot of cambium is becoming active and they are cutting secondary tissues in a ring-like fashion. Okay. So in this what will happen actually, normally in root the vascular bundle arrangement is quite different. Okay, you have definitely you have epidermis and then endodermis, pericycle, all those things you will have. Okay, inside it, but the vascular bundle is not conjoint, it is radial. Okay, you will see xylem and phloem in patches. Suppose two patches are there in this Beta vulgaris. Okay, you will have xylem and you will have phloem. This is the xylem and this is the phloem. Okay, so this is a diarch condition. Okay, two patches are there and the rest of the tissues are also these are also known as conjunctive tissue. Okay, they are parenchymatous in nature. So I have told you before that how secondary growth formation takes place. So what happens below phloem? Okay, below phloem some of the tissues start behaving like meristematic. Okay, so two cambium strands will be formed. Then some regions in the pericycle, in the protoxylem region, because in this root you will have exarch condition. Okay, it will be radial arrangement. So some tissues in the pericycle region, they will also become meristematic. This tissue will extend and it will join together and it will have xylem on the inner side. It will encircle xylem on the inner side, leaving phloem on the outer side. So this type of structure you will have later. In the center, suppose this is the cambium ring and this is forming xylem, encircling xylem here, and phloem is on the outer side. This is the cambium ring. Okay. Now in this case, this cambium, what it will do, this will start cutting. Now this cambium ring will start cutting xylem on the inner side and phloem on the outer side. Now in this case also it will not behave normally, because in normal condition this cambium ring was also forming a continuous layer of xylem and phloem. Okay, but this is not in the case of this Beta vulgaris. What will happen? They will also cut they will also have parenchymatous tissue. Okay, in the they will be separated by parenchymatous tissue. Okay, so this is the phloem and this is the xylem and this is the parenchyma. So what later what it will appear, that in the center you have two protoxylem patches, two xylem patches, okay, with protoxylem on the outer side, and then ring of vascular tissues. Okay, so rings of vascular tissue, because in between two vascular tissue, parenchymatous tissues are there. So now they appear as discrete patches. If suppose sclerenchyma would have been there, then it would have been blended with the xylem, because xylem is also lignified and sclerenchyma is also lignified. They would have blended together, but because of the presence of parenchyma, they are all appearing as discrete patches. Now in this root also, because see, it's a storage root, so definitely the lignified tissues are very scanty. Okay, okay, the lignified tissues are not as much. Like in xylem, you don't have that much of lignification, and mainly the parenchymatous tissues. Okay, parenchyma is more in vascular region. See, in vascular bundles also you have parenchyma, because xylem also have xylem parenchyma, phloem also have phloem parenchyma. Okay, but when they are performing secondary tissue formation, the amount of parenchyma will be more as compared to rest of the tissues, because they are basically modified for storage, and parenchyma is the one which will be utilized in storage, because if they are having lot of tracheids and vessel, there is no the function of that tissue is not fulfilled. So more parenchymatous tissue will be there in the secondary vascular regions also. So in this way, now again one more cambium will arise in the parenchymatous region. Okay, again it will cut xylem and phloem same way, and again it will have more layers of vascular ring. Okay, again one more layer. In the center you will have two protoxylem. So in this way, lot of ring like structures will be emerged, and major bulk will be storage parenchyma. Okay, so more storage parenchyma will arise in those tissue parenchyma, and sugars will be stored. Sugar will be the storage product. So in this way you will have the formation of this dicot root. See, this is also the cellular diagram where you can see lot of rings of those cambia, lot of rings are produced. You can see rings of those tissue. They are producing layers of secondary tissues. Okay, xylem is there, then you will have phloem will be there. Okay, then this is the cellular diagram where you can see lot of storage parenchyma. So as compared to the secondary, as compared to the other vascular tissue, more amount of parenchyma will be there. Secondary xylem will be there, secondary phloem will also be definitely there, cambium will be there, but their structures will not be that much lignified. Now coming to another storage root, this is Ipomia batata, which is the sweet potato. See, it is the adventitious roots which have been modified. Okay, these adventitious or fibrous roots have been modified to give rise to the storage root. Okay, they also have some abnormal behavior. Normally in this case also you will have the patches of xylem and phloem. Okay, they will start as such. Okay, suppose xylem and this is phloem, and then cambium is arising, and this is encircling this this tissue. In this case also, with further development, this cambium will give rise to a primary xylem and secondary xylem, but will also contain large proportion of parenchyma. So in this case also, they are cutting xylem, they are cutting phloem. Okay, but large proportion is still parenchyma. Okay, so in this case also you will have rings of vascular bundles, but in that cases more parenchymatous tissues will be definitely there in that ring. Now again what will happen, some secondary cambia will arise in the parenchyma around individual vessels. Suppose this is the, suppose these in this vascular bundles you have lot of vessels, okay. You have lot of vessels. So around that vessels also some parenchyma will arise, okay, and they will form parenchymatous tissue. Okay, as I have told you when we were discussing about wood also, that in that parenchyma is also attached, and there are different ways how these parenchyma is attached. But as compared to wood, definitely the vessel elements will be very few. Lot of parenchyma will be there when we are talking about these storage tissue, and these extra parenchyma will also arise by some cambia which will arise around these vessels, and they will give rise to extra parenchyma tissue which will definitely serve as storage function. Now, this is also a transverse section of a root of Ipomia, where you can see lot of vessels are there. Okay, these vessels are surrounded by extra parenchyma. Okay, phloem is also definitely there. Okay, this is the cambia and this is giving rise to phloem, but many more parenchymatous tissue is there. See, in this cellular diagram also you can make out that around these vessels some more anomalous cambia is coming out, and which is giving rise to more amount of parenchyma. Okay. Now, I just want to discuss about Tinospora root. Although in this the behavior of cambia is normal, it is normally giving rise to secondary growth formation as in any root, but some sort of you can say it's not exactly anomaly, but if you talk in terms of different means parameters of a root, it is definitely an anomalous root. See, in this case, anomalous is that it is an aerial root. First anomaly, see, anomaly means something which is not normal. So definitely if the root is not arising from its normal location, it will be considered as an anomalous structure. So in Tinospora it is also aerial, so it is anomalous. It is performing photosynthesis. So this is also an anomalous behavior. It will have chlorenchyma in its cells, because see, it is an aerial root, so definitely it is getting light and it is also serving the purpose of photosynthesis. So this is also anomalous behavior. In this, when we will when we will see the section, you will see a multi-seriate medullary rays are there. Okay, so normally in the xylem and phloem, okay, you see uniseriate or very thin multi rays are there. Okay, these are medullary rays. Okay, but in Tinospora you will see multiseriate. Okay, I will also show you the section. So when you will see the Tinospora root, see in root, as we know that we have studied before also, that previous slides also, that it will have xylem and phloem in patches. Okay, and this will encircle, and when the cambia will form, it will encircle it like that, and then you will have a continuous xylem. Okay, and in the center you will see the primary xylem. In the center you will have primary xylem, and then you will have phloem. In this you will see primary phloem will be in patches. Okay, it will be crushed, it will be in patches. Okay, this will be the primary xylem, because see, it's in the center, so it is not getting crushed. So you will be able to see the primary xylem in the case of root, and then you will have secondary xylem, then you will have secondary phloem, and then primary phloem. Okay, when you will see the structure of a in this way, this Tinospora root will also form secondary structure, but when you will observe it, you will see that some sort of discrete patches. It will appear that it's a dicot stem. It will appear, but it's actually not a dicot stem. You will see discrete patches of these tissues. This is the secondary phloem. Okay. So although it has to be continuous, but it is appearing in the form of discrete patches because of the presence of wide medullary rays. Okay, you can see here that in between these xylem and phloem patches, okay, secondary phloem and this is secondary xylem. You can see these are broad medullary rays, okay, elongated cells. So these broad medullary rays are filling the cap, and because of it a discrete patches of this secondary tissue is appearing, and it is not appearing in the form of continuous band, it should appear. So these are the medullary rays. Okay, now when we can see the transverse section of this stem, you can see lot of discrete patches, small amount of primary phloem you can see, which is almost getting crushed. It's a root also, so it will also develop cork, and cork cambium will give rise to cork on the outer side and secondary cortex on the inner side. Maybe sometimes it is secondary cortex is formed, sometimes it may not be there also. Then you have secondary xylem, and then secondary xylem and secondary phloem. So you can see discrete patches, and you can clearly see primary xylem, where this will be the protoxylem and this will be the metaxylem. Now this is the slide of a Tinospora root which I have prepared, where you can clearly see the cellular structure. You can see this is all secondary xylem. See, you can secondary xylem. You can clearly see that so much big vessels are there in the secondary tissues. That this figure I have also shown you previously. So this is about Tinospora root. Okay, so big vessels are there. Okay, you can easily make out the difference between these vessels. This is small primary vessels are there in the primary xylem. So this is protoxylem. In that also you have metaxylem, the bigger vessels. In this primary xylem also, the bigger one is in towards the center, and the smaller one is on the peripheral side, okay, on the outer side. So this is protoxylem and this is metaxylem. Okay, in the center some small pitch region could be there. Now you can see broad medullary rays. See, these are all medullary rays. Now they are not actually broad, it's not a normal feature that they are so broad, but in Tinospora root you can see broad medullary rays. Medullary rays, okay. So this type of structure you will see in Tinospora. So with this I will end up the discussion about secondary structures, the anomalous feature which are found after secondary growth, and I hope this all would have cleared some of your concept regarding anomalous structure and how they originate, and in which plant species particularly. I have tried to take examples of different species in which those secondary structures or anomalous structures are found. So it will help you in better understanding. Now you can also do some sort of work from your side also, that you can also try to analyze some plant. If it is possible, you can take those plants, and many examples I have told you. If those plants are available nearby, like Nyctanthus might be there, or many other species could be there. Dracaena could also be easily available in houses also. So that those plants you can take, and if possible, if it's there in your lab and if you want to test them, you can cut sections and you can stain them and you can analyze. Without staining also, if some stain is not available, you can without staining also you can try to analyze, try to cut very thin section and try to analyze those anomalous behavior. Okay, so for this whole topic is ended here. So thank you.